Hello friends, today is a good day to be alive, isn't it? Welcome to my pessimistic guide to anti-aging research, episode 9. Today we will be talking about epigenetic control and its relation to aging. DNA is a library that contains actionable and often mutually exclusive information about essentially everything, so it cannot be released all at once. The way this information is used is determined by epigenetic control. In relation to DNA, epigenetic control performs a function of a straitjacket, which is generally very restrictive and at the same time allows a degree of freedom in certain areas. Through selective activation and silencing of genes, such an arrangement makes possible creation of a variety of cell types, even though they all are genetically identical. So, essentially it is a relationship similar to hardware, or rather wetware, versus software. Here comes slightly boring part, but bear with me, it will last only about 20-25 seconds. The epigenetic control is executed through changing the way DNA is modified and packaged. The gene-specific control is maintained by methylation of cytosine in the promoters of the genes and resulting modulation of their responsiveness to transcription factors. Greater promoter methylation roughly translates into lower activity of the gene in question. Larger scale control is executed through combined action of DNA methylation and modification of proteins, histones, which can permanently or semi-permanently block or open access to large regions, thus shutting down or activating groups of genes. And this is all we need to know for the purposes of this mini lecture. Aging-related changes of epigenetic control are bidirectional. Some genes are hypermethylated, or in other words silenced, and some regions become hypermethylated and therefore active, undesirably so. There are some very consistent trends in DNA methylation that allow a remarkably precise determination of biological age, but there is also a strong random component. For example, while epigenetic profiles of young monozygotic twins are almost indistinguishable, the differences between older twins are rather significant. Also, it has been shown that within the twin pairs, the individuals with greater epigenetic age tend to die sooner. The overall aging dynamic reflects growth of disorder. In young humans and mice, the patterns of DNA methylation are nice and tight, while the older individuals show much greater scattering. This is a clear indication of entropy that slowly eats away the established order of interactions between all genes, whether mutated or not, which our existence depends on. In some ways, the epigenetic aspect of aging scares me more than mutations. I can compare mutations to honest-to-goodness straight-shooting bullets with an ambiguous and permanent local impacts. In contrast, deterioration of preset epigenetic programming seems to me far more insidious. I have a vision of aging-related changes in epigenetic control as a semi-liquid tide of entropy that creeps over the entire genome and spares nothing. Now, let's address the question of relative importance of mutations and epigenetic decay to aging. This question can be answered through serial cloning, using somatic cells from an adult animal for several generations. In early experiments, cloning failed after a second or a third generation, and cloned animals showed various abnormalities, including shorter lifespans. These results seem to confirm that accumulation of mutations is one of the primary causes of aging. However, recent groundbreaking experiments are forcing a drastic revision of this notion. Enter Wakayama's group from the Riken Center for Developmental Biology in Japan. There are many other groups with similar findings, but Wakayama's results are the most impressive. They serially cloned mice for 25 generations, and all generations were healthy and had normal lifespans. because serial clones were made from somatic cells of 3-month-old animals, and because the last clones had normal lifespans, it brings the cumulative lifespan to more than 9 years. I am almost sure that they discontinued the experiment simply because it was no longer exciting. Keep in mind that mutation rates in mice are much higher than in humans, and that individual mouse cells accumulate just as many mutations as human cells. If this is not sensational, I don't know what is. If we accept the conventional understanding of the role of somatic mutations in limiting lifespan, the increasing mutational burden should have prevented this nonsense of serial cloning from continuing for as long as it did. At a minimum, we will have to make a distinction between the lifespan limiting effects of mutations and the ability of individual somatic cells to become the origin of a healthy clone. Mutations can cause aging through creating a mosaic of variably, albeit slightly, compromised somatic cells, because this effect does not require cripplingly large mutation loads. In other words, we need to define mutation load in relative terms. It is high enough to cause growing dysfunction within organs and tissues, and yet it is low enough for at least some somatic cells to retain the ability to generate a perfectly normal clone. There are some additional developments as well. Another blow to precedence of mutations comes from the revolutionary discovery that any somatic cell can be reprogrammed, which involves complete erasure of the existing epigenetic patterns, into a stem cell using few transcription factors. Coincidentally, reprogramming is also accompanied with rejuvenations. Even the somatic cells of centenarians can be rejuvenated and reprogrammed into fully functional stem cells. There are also very encouraging reports on successful in vivo rejuvenation through epigenetic reprogramming. So, figuratively speaking, of this old body of ours, we may be looking not at a hopelessly decayed piece of wetware, but at an only slightly used one that can be made like new by wiping off the old and installing a new operating system. Sure, we have a few malfunctioning circuits and damaged segments on our hard drive here and there. So what? It does not mean that we are not fully operational. Intriguing, isn't it? Can it be done? I don't know. But it is probably worth mentioning that many traditional lifespan extending interventions are known to exert a certain influence on epigenetic control. Caloric restriction, for example, reduces the rate of epigenetic drift and rewinds the epigenetic clock to a younger age. Or resveratrol. It is hailed mostly for its antioxidant properties, but it is also known to act via DNA methylation and histone modification. So, to summarize, the decay of epigenetic control may be a realistic candidate for the most proximal mechanism of aging, based on the following lines of evidence. First, there is a correlation between the rate of epigenetic drift and lifespan across mammalian species. Second, we have a proven possibility of superserial cloning. Third, we also have a proven possibility of rejuvenation of any somatic cell, including cells from centenarians, by epigenetic reprogramming. And also we have limited evidence that at least partial rejuvenation through epigenetic reprogramming is possible in vivo. And the main tentative conclusion is that in terms of accumulation of mutations, we are probably too young to die at the age of 80, or even at the age of 120, or maybe even beyond. And on this optimistic note, I will excuse myself. See you next time.